Okay, welcome back, everybody. We're going to continue on in chapter 4, which is the survey of the prokaryotic cells. We've talked about external features and a little bit about the cell envelope of prokaryotes. We want to continue our survey of the cellular structures by examining some of the internal features, starting off with the cytoplasm. And we're all familiar kind of with what that is. Because if you think about eukaryotic cells, basically everything inside the cell membrane that contains the organelles is within the cell cytoplasm. Well, prokaryotic cells, like bacteria, also have a watery cytoplasm that contains a variety of chemicals, and they list a few of them here, different types of carbohydrates and peptides and proteins and amino acids. So there's a whole host of chemicals within the cytoplasm, but the majority of what's there is water. We also know that prokaryotic cells are unique in the sense that they lack a membrane-bound nucleus that would contain DNA and protein, like in eukaryotic cells. So that doesn't mean, of course, that they don't have DNA or that they don't have a chromosome, because they do. So in prokaryotes, you have a single circular piece of DNA, of course being double-stranded. Unlike eukaryotic cells that have numerous chromosomes, bacteria just have one, and it's circular, which is kind of interesting because prokaryotic cells, as we'll see in a few moments, have the unique ability of replicating that circular piece of DNA and passing that on to the daughter cells in a process called binary fission. So more on that in an upcoming chapter. In addition to the single circular piece of double-stranded DNA, many bacterial cells also have what are called plasmids, which are small, tiny pieces of what we would term extrachromosomal DNA. It's, of course, also double-stranded. And as we'll learn about in upcoming chapters, these plasmids can provide a lot of benefit to bacteria. For example, being resistant to certain types of antibiotics or drugs is imparted by the presence of specific types of plasmids that have basically the genetic blueprint to negate the effect of the antibiotic or the drug. So more on this coming up, but there's been a lot of research done on plasmids in bacteria. As the third bullet alludes to, a lot of research has been performed looking at how can we manipulate plasmids of bacteria, how can we insert certain types of desirable genes within the plasmid itself, and then as the bacterial cell reproduces, undergoes cell division, those subsequent daughter cells, if you will, would then also have presumably those same abilities to do whatever the plasmid provides. So more on that coming up in an upcoming chapter. But plasmids are very cool pieces of extrachromosomal DNA. Ribosomes, we said, was really the only commonality, if you will, between prokaryotic and eukaryotic cells, other than maybe the cell membrane. This organelle, you remember hopefully from bio and A&P, is instrumental in the assembly of proteins. And like eukaryotic ribosomes, prokaryotic ribosomes also have a larger and a smaller subunit, which fit together, like you see in the diagram here. These particular oval structures, if you will, within the model depict where transfer RNA molecules actually fit into the ribosome. If you remember from A&P, it's a process called translation that involves the bringing in, if you will, of amino acids by transfer RNA molecules. Those eventually get linked together to form the protein. And so these are simply sites for those transfer RNA molecules. And in bacteria, we have this upper 50S subunit and the lower 30S subunit. And I've always been puzzled as to why together they don't make up an 80S subunit, 50 plus 30. but it's referred to the entire ribosome as a 70S. And so when you compare, again, the ribosome of a prokaryotic cell with that of a eukaryote, there are some differences with respect to size of these organelles, which by the way, are primarily a ribosomal RNA or rRNA. That's another kind of RNA in addition to messenger and transfer. So ribosomal RNA goes into the construction really of the ribosome itself. In addition, some bacteria have internal structures like inclusions and granules, which they use to sometimes store nutrients when things get sort of unavailable, if you will, within the environment. Those bacteria can utilize those stored bits of protein and carbohydrate and lipid and other chemicals. And they often store those, of course, in the structure here, the compartment called an inclusion. So these are what are shown here on the left-hand side, these pink structures. This is depicting a very interesting bacteria that is storing iron in its granules. To reach you on your cellular device. And in this case, it allows for this cell to get a better sense of the magnetic field within the Earth. It's fascinating to study this. Who would ever think that we would have bacteria that could orient themselves based upon the Earth's magnetic field? So this is a taxus. A magnetotactic bacterium would have those iron ore chemicals in their organelles and could detect the Earth's magnetic field. As to how they respond to that or what benefit it provides, I don't really know, but it would be fascinating to do some additional research on what does that provide? What benefit does that provide these bacteria being able to orient to the Earth's magnetic field? Fascinating stuff. Within the cytoplasm, and this applies actually to eukaryotes to some extent as well, there is an internal cytoskeleton that can provide internal rigidity to the cell. Now, even though it's 80% water, these actin filaments, which are contractile filaments, as shown here in the stained slide, these yellow structures, which look sort of like DNA, but it's not. These are actin filaments. You've heard of actin maybe as being part of the protein found in skeletal muscle, right? And in other types of muscle as well. It's a very similar compound, but in this case, the actin is providing internal rigidity, internal support, I guess. Rigidity is not a good term to use because it's not a rigid structure, but it provides internal support to the cell. We know from an earlier chapter when we talked about the endospore stain that some bacteria, specifically those within the genus Bacillus and Clostridium, have the unique ability to form these dormant structures called endospores, typically when the nutrient supplies really become depleted or when there is an environmental change, like a significant temperature shift in the environment. Those sorts of factors can induce the formation of endospores. So when we talk about and look at cells in the microbiology lab, we're usually talking about and looking at vegetative cells. Those are the normal metabolizing forms. But in the case of Bacillus and Clostridium, again, when conditions turn harsh, the vegetative cell can form an endospore within itself. And that endospore is able to basically survive until conditions improve, and then they will germinate into vegetative cells. And this process of sporulation or formation of the endospores can take a bit of time, as the slide indicates here, upwards of 6 to 12 hours. So it's not something that happens super quickly. But it's a fascinating structure, the endospore. And the reason that it can remain so resistant is often attributed to the production of what's referred to as dipicolinic acid within the vegetative cell. Actually, it's within the endospore, which is forming within the vegetative cell, as the next slide will talk about in the link video. But the dipicolinic acid seems to be really critical, because when that forms within the endospore, then what we find occurs is calcium moves in and water moves out of the endospore. So having this very desiccated state is critical to the formation of endospores. Now, how long can an endospore sit there in this dormant state? Well, as it mentions here, a long, long time. Upwards of 200 million years. Hard to believe, but they have actually found endospores that they've been able to basically to germinate, if you will, back into vegetative cells, even though they've been sitting there dormant for that long a period of time. It's really phenomenal to think about that. And so being so resistant means that if we want to ensure sterile conditions, we would have to apply some pretty significant temperature and pressure. And that's, of course, what's described here in the last bullet. When we go to sterilize media, for example, we have to employ the use of what's called an autoclave. There are other instruments we can use as well, and we'll talk more about that in another upcoming chapter. But the autoclave is sort of the standard that most labs use, a dental office, the hospital, and so forth will have autoclaves. And so the autoclave imparts a pressure of a minimum of 15 pounds per square inch at a temperature of 121 Celsius, is generally the standard, for 15 to 20 minutes to ensure sterility, to ensure that we're killing not only the vegetative cells, but these very resistant endospores. So here's a figure that's kind of describing the process by which endospores are forming, whether it's Bacillus or Clostridium. And I'm going to just ask you to kind of look through these various steps as we go clockwise from the top. Also, this YouTube video will take you to a minute 30-second animation. And there they talk a little bit more about the various steps, the importance of the dipicolinic acid, the influx of the calcium, and the exit of the water, again, to provide this very desiccated state for the spore. But as you can see from the diagram here, the endospore forms within the vegetative cell. We'll be doing an endospore stain in lab coming up in about 3 weeks. Okay, let's shift gears a little bit and talk about morphology of bacteria. And I think it's important to note that the vast majority of cells that you're going to encounter in the laboratory, or even out in the real world, if you will take on generally one of these three morphologies or shapes. There's the round spherical coccus, the rod-shaped bacillus, and the spiral-shaped, helical-shaped spirillum. So these terms in yellow are in purple are depicting morphologies, shapes that we attribute to most bacteria. There are some exceptions to the rule that we'll talk more about. It's not just falling into one of these three categories, but the vast majority do. And on this slide, we can see some what appear to be mostly scanning electron micrographs, except for this lower right-hand photo. This appears to be from a compound light microscope. But here we see coccus, the spherical morphology. These happen to be in a cluster, so we would add the term staphylo in front of coccus to better describe these. Staphylococcus means grape-like clusters. Here's bacillus morphology. Looks like hot dogs kind of linked together in some respects. Here's a variation of the bacillus. It's referred to as Vibrio. Vibrio cholerae is probably the most common type of Vibrio bacteria. It causes cholera, which is a waterborne intestinal disease that kills millions of people every year. They get terrible dysentery as a result of cholera, ingesting the Vibrio bacterium. But what you can notice here, of course, is that there's a flagellum sticking out the one end. Or in this case, we have actually a couple. Not all Vibrio have flagella, but some do. Here's spirillum, our spiral shaped. We won't really be working at all with spirillum. They are kind of cool looking, corkscrew shaped. And you can see again that some of them do possess flagella to assist in motility. Spirochetes are another shape, a unique shape that some species take on. They're a little more slender than the spirillum morphology is. So they're very slender kind of helices, helical-like shapes. And then certain types of bacteria may even exhibit branching morphologies. You can see it would appear to be some coccus, but there's more branches also. In addition to having this kind of streptococcus morphology, you have branching, and so that's depicted here in the lower right-hand corner. We will again talk mostly about coccus and bacillus morphologies. It is important to note that there can be variation within species in terms of their shape and even their size sometimes, which makes identification sometimes kind of tricky. So when we say the term polymorphic, this means many shapes. And so some kinds of bacteria are a little more known for their pleomorphic abilities, their abilities to exist in a variety of shapes and sizes. And so when you go to work on your unknown, I think you need to be kind of open-minded in the sense that while you may see your suspected bacterium as being listed as bacillus shaped or coccus shaped or whatever the case may be, if you see some other shapes when you do your staining of your unknown, don't just assume necessarily, hopefully, that your culture is contaminated with other kinds of bacteria with different morphologies. It could just simply be that the bacterium that you're working with may be slightly polymorphic. So just, again, something to keep in the back of your mind. So in addition to having, the overall shape of, say, rod-shaped cells like bacillus are, or spherical-shaped cocci, these can occur in different arrangements. And that's what's kind of depicted on this slide. The fact that we can have some cocci forming what are called, well, diplococci is the proper term. I was thinking of the term pair. They occur in a pair. So 2 diplococci, as opposed to a long string or chain, and we would use the prefix strepto to define that particular morphology. Earlier, we talked about staphylococci, which were grape-like clusters, sort of what we see down here in the lower part of the figure. And a lot of times, the morphology is dependent upon how the cells divide. I'm not going to get into a lot of this, but if they divide in perpendicular planes as opposed to multiple planes, it can result in sort of a random cluster of cocci like we have here in this case of Staphylococcus, because the cells are in a variety of planes as it relates to how they orient to one another, the individual cells. So just something to be cognizant of. A variety of shapes, a variety of divisional processes that can result in unique shapes. This is just another slide that kind of has us hopefully take a step back and realize just how small, again, some of these bacteria are, like E. coli or Staphylococci, compared to, say, a red blood cell or a pollen grain. So again, we often need to use specific units, right? Micrometers in the case of the funny to better gauge the size of these cells, or as it depicts here, Ebola. Viruses are even smaller than bacteria generally. So when we go to study how prokaryotes are classified, keeping in mind again that we're discussing here two of the three domains, archaea and bacteria, most microbiologists, at least those that would be working in a laboratory, say a college lab teaching, we would be interested in these three classification terms or categories, I guess. If you were working at a higher level, governmental or university lab, you might delve into number 4 and number 5 in an attempt to better understand how bacteria are classified, how they're related to one another. But generally, we're going to be looking at these three, at least in the lab that we have. And one important group of books that we're going to be looking at is referred to as Bergies Manual. There's actually a cluster of manuals, but it came out initially as a single text back in the 1920s. And this was the fella, Bergie, obviously is his name, that published the very first book of systemic bacteriology. It has since undergone a whole host of revisions. It's been expanded as we've learned more about taxonomic relationships. As our technology has increased, we've begun to see that what assumptions we may have made decades ago needs to be revamped. We learn more about the genetics, for example, of certain kinds of prokaryotes, means that we may have initially classified them taxonomically incorrectly. So this is a process that is ongoing constantly. But we will be using, again, Bergies Manual quite a bit. You'll be using it as you begin to hopefully trace the identity of your unknown in lab. Most microbiologists, most bacteriologists think of the three domains that exist today phylogenetically. They look at it or examine it in this kind of method. These terms that you're seeing here in blue and green and red, I'm not going to really ask you any of these. Now we'll talk more about some of these terms coming up in the next chapter. So you've heard of plants and animals and fungi, maybe in protozoa. In fact, we examined those in lab not too long ago. And you've heard of cyanobacteria. But a lot of these other ones are terms specific to either the domain bacteria or the domain eukarya or to the domain archaea, obviously. But what I want you to get out of this slide is the fact that the domain archaea, even though they're prokaryotic, notice that both the archaeans and the eukaryotes share some common lineage, while the bacteria domain and all the organisms found within it are sort of out on their own. They evolved in took a slightly different or a significantly different path than did the archaea and the eukarya. They eventually diverged, okay? They split here, but they do share some commonality, some evolutionary phylogenetic history, and we're going to allude to that a little bit later. So don't, again, get caught up in all of this terminology, but try to get a sense of how these three domains sort of stack up against one another with regard to evolutionary, general overall evolutionary processes here. So back to Bergies Manual for just a moment. What Bergies generally does is it takes the prokaryotes, and breaks them up into 5 major subgroups. And within each subgroup, we have various phyla. Now, this is a plural term. If I'm talking about one, I would use the term phylum. Okay. I don't want you to get all worried about having to memorize table 4.3 in your book. That's not the purpose of the next two slides, but rather is to give you some sense of the diversity within these 5 major groups. And what the book does is basically color codes the 5. There's a yellow, a green, a purple, and on the next page you'll see kind of a violet and a light blue part to the table. So we can see here the domain Archaea in yellow. The remaining groups here in the various colored areas of the table are going to contain the bacteria domain. So the Archaea domain, kind of small. The bacterial domain, much larger in terms of diversity. Look at all the different phyla terms and names. And if you want to read over, the description that they provide here in the first line of each of the boxes, you can see what makes these, bacteria in this case kind of fall in the same group, or why they've been categorized within this particular category. But we're not going to get all caught up in the taxonomy. But there is a taxonomic nomenclature that would be discussed and used among microbiologists as they talk about these various prokaryotes. What the book also does in table 4.4 is it focuses on some of the more important bacteria as it relates to infection and disease. So this is irrespective of phylogenetic or evolutionary relatedness. This is looking strictly at morphology, in other words, shape. That's what this word phenotype kind of means also. Phenotypic characteristics or qualities are used. In other words, how do the cells appear under the microscope? And what they're doing here basically is they're saying, in the upper part of this table, here are the gram-positive cells that we know contribute to disease or infection. Here are the gram-negative bacteria that we know contribute to infection and disease. And some of these, as you peruse through the table, some of these bacteria you're going to have heard of before: Staphylococcus, staph infection, Streptococcus. Here's Clostridium and Bacillus, spore formers, right? Which can contribute to gangrene and botulism and something called anthrax. Here's bacteria that cause acne problems, tuberculosis. These are all gram-positives. And then as we go down into the lower half of the table, we see some others that may again look familiar to you. Gonorrhea, Neisseria is the genus. Meningitis. Here's plague. Yersinia pestis causes plague. Salmonella, you've heard of salmonella infections, like lettuce or spinach sometimes has salmonella that gets into it, and they have to redo a big recall, right? Here's cholera, Vibrio, the genus, and so on and so forth. So just take a few minutes and kind of look through this table of some of the medically important pathogens. Well, the term species, if you recall from an earlier discussion, is sort of the most specific term we could use in the taxonomic process of naming and categorizing organisms. We know, for example, that we have a scientific name. Our genus is Homo. Our species is Sapiens, Homo sapiens. Well, bacteria, as you've already probably figured out, also have genus and species terms as well. Escherichia coli. Coli is the species. Bacillus subtilis. Subtilis is the species. We haven't worked with that yet, but we will soon. In lab this week, we worked with Serratia marcescens. Marcescens is the species. So what makes a species a species? Well, if cells have at least 97% of their genome, their genetics, and their rRNA, which is pretty much the ribosome, if they share at least that much between two cells, they would be considered of the same species. Okay, that's technically what we've adopted, that particular definition. However, to muddy the waters a little bit, you can have what are defined as strains or varieties of species. So they share, 97% of the genome and rRNA, but sometimes you can have slight phenotypic differences between cells. And in this streak plate that we're seeing at the bottom of this slide, we can see some red colonies and some white colonies. And if you looked at that, you would probably certainly assume that we're looking at two different species here, because one's definitely red and one's definitely not red, it's white. The fact of the matter is, we're looking at Serratia marcescens, which can, depending upon the incubating temperature, form not so much red colonies, they're actually a bright pink, as well as white or cream-shaped, cream-colored colonies. So it's the same species, Serratia marcescens, marcescens is the species, but we can have two different strains or varieties depending upon what temperature we incubate Serratia at. So it boils down to certain genes getting turned on or turned off, color genes, if you will, that results in the pink or no color, it's white. So that's kind of interesting. Okay, I'm going to actually stop the Zoom lecture right now. And when we pick up and wrap up chapter 4, we'll focus on some interesting, unique prokaryotes. So we'll pick up where we left off.